We're starting the recording. So, hello. So today we're gonna try to cover eukaryotic gene regulation. We talked about prokaryotes last week, and that was the operon system. Remember that in prokaryotes, in bacteria, all the genes that are needed for a particular pathway are all together, one after another, in one set of, in a structure that we call the operon, that includes a promoter and includes all the genes that are needed. So everything comes in a package, like a meal number 7 at McDonald's. You ask for a number and they give you everything you need. In eukaryotes, this is slightly different. The genes for a particular pathway or anything, they are all over the place. And there are many points of control, that places where you can turn the genes on and off. And I want everybody to remember that most eukaryotes are multicellular, and the cells are much bigger, and as such, they need to coordinate a whole body system. All the cells have to communicate with each other to be able to take the body and allow the body to perform perfectly. On top of that, about the coordination of multicellular organisms, you have also to deal with the problem that, or not the problem, but the great advantage of that, since we are multicellular, we have cell specialization, and we have multiple structures that are going to do very specialized things. And for that, you're going to have genes that are turned on and off. And that's an important consideration, that throughout our lives, some genes are coming on, some genes are going off all the time. And your body has to coordinate that. So please remember then that in prokaryotes, there is absolutely no coordination. It's all systems on or off. Cells are small, so regulation is at the gene level. If you need something, you turn your genes on. You produce the enzymes needed. If you don't need them, the genes are off, and everything else is recycled to maximize the space available in prokaryotic cells. Also remember that bacteria must respond very quickly to their environment, pretty much like everybody else, but since they are unicellular and small, they have an added pressure for that. When you are dealing with eukaryotes, there is a difference. We have bigger cells again, we are multicellular. So gene regulation is more independent. You don't need to regulate all the genes at once. And your body has to, every cell of your body has to kind of listen to the signals that are being sent from other cells, and has to do something like a triage section, where you have to kind of listen and decide what is the best thing to do at a given moment. And sometimes you have to your cells have to deal with conflicting messages, and they do basically, using those messages, they prioritize, and that's how they are able to turn genes on and off. So that's what I want to go over now. It's gonna feel a lot like a laundry list, because basically gene expression really means starting with DNA, where the code for your genes are. Genes are going to be expressed if you can produce a protein or something that is going to happen in your body. So from transcription, or even before transcription, from the moment that the DNA is wrapped around histones, all the way until a protein is produced and functional, and that's important, functional to do its job, there are many places where you can stop the process. So gene regulation does not have to happen at the DNA level, or does not have to happen at the very end. It can happen in a huge number of places, and that's what I'm going to go over now, all the different places where this can happen. This is a nice diagram that you have in your textbook that basically has all these possible places, and in the room also I have a nice poster, the way I understand it. So I'm just going to put all this here. No need to stop right now, but the different places. And we are going to start one by one, covering each one of them or mentioning a few things. So first level of regulation. Even before transcription happens, basically you need to transcribe the gene, and for that the gene has to be available to be transcribed. If you recall, all your DNA is coiled around histones, and then these histones, the DNA coiled around histones, that structure is called a nucleosome, and that's a fundamental unit of the wrapping, the coiling of the DNA. And then these nucleosomes wrap around them each other, and then they keep on wrapping. It's a super coiling system, basically, and you end up doing the chromosomes. The only time that you are going to be able to transcribe anything out of your DNA is if your DNA is uncoiled. So the first issue that we have here is the first level is what we call histone acetylation. And acetylation is the addition of an acetyl group, COCH3, to our histones. So in my model of DNA that I have here, so these are our histones, and the DNA is coiled. When the DNA is coiled around the histones, there is no transcription there. So your genes, any gene that is in this DNA is technically off. However, if I attach an acetyl group, and there is an enzyme that specializes in doing that, if I attach an acetyl group to the histones, basically that changes the charge of the histones, and the DNA that is negatively charged basically is kind of repelled, unravels, and now these genes here are available for transcription. So histone acetylation, critical component to allow for transcription to happen. When these genes are transcribed and you have a messenger RNA, then you don't need it anymore. There is another enzyme actually that removes these acetyl groups, and immediately the DNA, like a spring coil, goes back and recoils around the histones. So first level of regulation, histone acetylation, very critical component. We have learned that it's very, very important. So here you have non-acetylated histones, histones without the acetyl group. Everything is tightly coiled. And here you have a histone acetylation, and you are able to see the pieces of DNA that can be transcribed. In the last unit, we talked about bar bodies and X chromosome inactivation. Well, hello, this is how it happens. It's a combination of acetylation or deacetylation. You remove the acetyl group, so all the DNA is tightly packed. And as we will see the next one, it also is something called DNA methylation. So these two, histone acetylation and DNA methylation, are going to be involved in the production of those bar bodies that we talked before. So acetylation of histones, basically because you acetylate and you release the DNA, that is going to turn your genes on. That's the basic premise there, and allows for transcription to occur. The next one that we have is also at the level here before we even have transcription, and this is called DNA methylation. And it's the addition of methyl groups, CH3 groups, to DNA, specifically to the cytosines, the nitrogen base called cytosine in the DNA. If you look over here, you have a regular cytosine. Notice in carbon number 5, cytosine has a hydrogen. When you have methylation, instead of a hydrogen attached to carbon number 5, oops, my loop moved, you are going to have a CH3 group attached to it, a methyl group. Now here on the side, you have a nice DNA, and if you can see over here, you have methyl, and methyl groups all over the place. This is basically what DNA methylation is. So, how does this work? Yeah, there is an enzyme that is going to attach those methyl groups to the DNA, and it's going to work two ways here. First, if the methyl group is attached to where a promoter region is in our gene, remember that transcription factors are going to attach to the promoter. So if I have my DNA here, but I have some methyl groups attach right here where I have my promoter region, the transcription factors basically are not going to be able to attach. Remember, all this, there is no magic, is all about shape. So the methyl groups are not going to allow the transcription factors to attach, and there is no transcription, so genes are off. The other possibility with the methyl groups, instead of being attached to the promoter region, they can be attached anywhere along the gene, actually. Just like you see here, as long as there is a cytosine, it's a group of cytosines together, they are going to attach there. So if I'm transcription factors, bingo, they attach there nicely. After transcription factors attach, here comes RNA polymerase attaches, and RNA polymerase starts transcribing the DNA nicely until it gets to one of these methyl groups. And that is like a nice roadblock there. Imagine putting some rocks or something in the tracks of the metro. It cannot move through. No trains can go through. So that's what this does. Basically blocks transcription, either by blocking the attachment of transcription factors or by blocking the movement of RNA polymerase through the DNA. So simple. Now this particular issue of blocking DNA methylation, of blocking the genes with methyl groups has, and also, for the most part, this is a new field of research that is called epigenetics. Epi means on top, so it's on top of your genes. And what it means, your DNA sequence has not changed and doesn't change much unless you have mutations throughout your life. Your DNA sequence has not changed, but by adding methyl groups, you can block genes and you can actually change your phenotypic expression, because even though you have the sequence, you have the genes, they are technically blocked by these methyl groups. A number of studies have been done in twins, identical twins, that sometimes they turn out very different. Not only physically, but also emotionally, psychologically. And what they have been discovered now, when they compare the DNA of these identical twins, the sequences are the same, no change whatsoever. But the methylation of individual genes has or is different between these twins. It's fascinating, because basically that means that actually your environment can change the phenotypic expression of your genes beyond what we thought at the beginning. Kind of Lamarck was actually right. The funny thing about this is that whatever is happening to me right now in my body, whatever I'm doing, whatever genes are being methylated in my body, it's also happening in my eggs, in my ovaries, and the same with all of you. It's happening in your testicles, ovaries, all that stuff. So it's not only your somatic cells that are being methylated, but also your reproductive cells. So you can actually pass these changes to your offspring, at least for the next generation. Eventually, they get removed because there are some other things going on, but it has been shown that at least for the following generation, you are able to retain these methylated, these methyl marks, this methylation that turns off particular genes. It's a brand new field, about 5-6 years old. Now it's picking up speed. A lot of research, mainly in the social science, the psychology, is being done here, where things that they thought was just psychological changes, they are seeing that it's really, all those changes are being brought up because there are methylation marks, that's what they call methyl marks, methylation blocking particular genes in the brain of organisms. So fascinating. Just Google it, you're going to find a wealth of articles about this now, and it's a great place to start research. So methylation, so acetylation unravels the DNA, allows for transcription to occur. Methylation blocks particular genes, and that's the end of it. So that's the first level. So we haven't even had transcription. Now, let's assume that everything is good, your genes are available, your histones are acetylated, your DNA is unraveled, and we are going to have transcription. So here is where we have another level of change that we might have. If you look at the picture here, remember we have our promoter area where transcription factors attach, and the coding region of our DNA. After you have transcription factors attach, RNA polymerase attaches and transcribes the gene. Well, we have discovered too that upstream, since the genes are being read in this direction, upstream, going over there in the gene, there are other areas in our DNA that are called enhancer regions or enhancer sequences. The names are going to change a little bit. And these enhancer sequences are needed to basically activate the whole thing. We thought that, oh, just transcription factors is all you need. Well, a lot of genes need kind of a little kick, a little boost from these enhancer regions. And what they have discovered is that in these enhancer regions, there are some other proteins, other factors that are going to attach there. And these factors can either activate, like this one, that are going to activate the process, or sometimes they can block it. So it's like you're ready to go, but somebody before you determines that nothing is going to happen even you have everything ready to go. So if you look at this diagram, this is an activator that is going to allow things to work. And transcription factor, RNA polymerase ready, but it's only going to go and happen if an activator is attached to this enhancer sequence. And if you can see here in the DNA, it's a nice loop. All these proteins have to come together for RNA polymerase to be able to take off and transcribe. So very important. Here I have the same diagram. This is also in your textbook. I want you to be familiar with different diagrams representing the same principles because it's all visual. If you see it and you get it, you are in good shape. So same thing here. Notice we have the gene there that you need. There's the promoter with your data box. Here is where your transcription factors and RNA polymerase are going to attach. And notice that farther upstream in the DNA is where you have the enhancer region. Now, attached to this enhancer region, you can have these activators that can do two things. Can be activators that are going to increase the rate of transcription and facilitate the process, or you can have silencer or repressor proteins attached to it. Hey, repressor sounds the same as in prokaryotes. Easy way to remember. If you have a silencer or repressor protein attached there, guess what's going to happen? Absolutely nothing, even though everything else is ready to go here. So multiple levels, multiple things that need to be working together for transcription to occur. It's not as easy as we think. So, nice diagram here. So transcription factors, this is an ideal world. Here comes transcription factors, attach, RNA polymerase attaches there, ready to do transcription. And here the DNA that has had these nice activators attached to those enhancing sequences loops around, connects, and that is the let's go and let's do transcription signal and everything works. If instead of activators there, instead of activators attached to that enhancer region, we would have had a silencer protein or a repressor, this would have been totally blocked and there is no transcription. So another place where you can do this. So beautiful system, multiple places where you can regulate this. There are a couple of YouTube videos I have there. If you feel like it, watch them. There is nothing like a visual to see all this happening. It works beautifully. So we unravel the DNA. We had transcription, hopefully. Well, we still can stop it. If something happens in your body that decides that, ooh, we don't need that, we can stop it. And here, stop it or modify it. That's very, very important. So here comes the next level, that is at the level of when we decide to do the splicing. Alternative splicing also allows you to change or modify what you're going to produce on the spot. We have talked about this. A particular DNA is going to be spliced by SNRPs that build a spliceosome, and using the same original piece of DNA, you produce a messenger RNA, the primary transcript, but then due to alternative splicing, depending on how you cut and paste the different exons, you can have different proteins being produced. So at the last minute, you can actually, your body can change your mind about which proteins are going to need to be produced. So next, 5' cap and poly-A tail. We talked about this. The 5' cap is a GTP guanine triphosphate that is attached at the 5' end of your messenger RNA, the mature one. So there you have your phosphates. Imagine 3 big phosphate groups attached there. Changes the shape so nothing can attach to it. So enzymes that are trying to break it down are unable to start there. The poly-A tail at the end, the poly-A tail, the longer the tail, basically, the longer the life of the messenger RNA and the more protein is going to be produced. So this is a way of regulating the expression based on the quantity, determining the lifespan of your messenger RNA. So here I have a beautiful picture of the whole process. So you can, if you can visualize and keep this in your mind, you are going to understand pretty much how your DNA works. Notice here is your beautiful gene with the exons and introns. The promoter region with your data box where the transcription factors attach and RNA polymerase attaches there. And then these control elements, these enhancer regions where other specific proteins or factors have to attach to either superactivate or to turn off your gene. So when everything is in place, you have your primary messenger RNA transcript. You go through all the RNA processing with the spliceosomes, removing all those introns. You add your poly-A tail to determine the lifespan of your messenger RNA and your GTP cap, and you're ready to go. But this was another level where you could regulate the genes. Basically, remember, gene regulation is all about turning your genes on or off. Are you going to make a protein or not? So let's pretend that everything is working beautifully. We have transcription, we have splicing, we have our mature messenger RNA, and this mature messenger RNA is going to make it out of the nucleus through a nuclear pore because it has the cap and the poly-A tail. Ready to go. So here we go. We go into the cytoplasm. It's a wild world out there. You can hear it there. And now something new can happen to you. This is a dangerous place out there. So what's next is also a relatively new thing that is called RNA interference. RNA interference, and you can see it as RNA little I there. And again, this is something relatively new. So you have your beautiful messenger RNA that is coming out there. And what they have discovered is that there are 2 different types of RNA. One called small interfering RNA, siRNA, and another type that we call microRNA. These are small pieces of RNA, about 20 to 30, 40 nucleotides long. And what these pieces do, actually, they bind. So here this could be SI, small interfering, or it could be micro. We don't need to understand clearly all the distinctions between those two at this level, so don't get worked up about it. Either one will work for us. So these are small segments of messenger RNA that were transcribed previously. Actually, they have discovered that a lot of those introns that got cut up become these. So fascinating. Anyway, so here you have your small interfering RNA. These move to the cytoplasm from the nucleus. Remember, all RNA is transcribed in the nucleus. And then if the sequence here, if a sequence, U, C, C, G, 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 is somehow complementary to a section in your messenger RNA, they attach with hydrogen bonds. And when they attach, when they bind to this, it's basically a death tag. You're done. That messenger RNA has no hope because two things can happen. Thing number one, by attaching to this, remember that RNA is also a ribozyme, has catalytic properties. It's going to basically destroy the messenger RNA. It's going to break it up. Very sad. Messenger RNA broken up is going to destroy it. Or the second thing, by having this other piece of RNA attached to you, now it's kind of a double, it's not going to be able to be translated when it gets to the ribosome because physically something is blocking the codons there. So by small interfering RNA and microRNAs, two things can happen. Either they degrade, break down the messenger RNA when they attach, or they basically block translation because physically they're blocking the codons and tRNA and the ribosome cannot be built around them. So, small interfering RNA and microRNAs basically cause gene silencing, basically turn the gene off. All this work, the cells have done all this and then all of a sudden they gave the instruction that is not needed. Well, here is another place where they can regulate their genes. Incidentally, this is a new thing, relatively new thing, just a couple of years old, and a number of biotech companies are trying to use this to cure some diseases. So what they are doing is making these small pieces of RNA and trying to inject them into specific, mostly for tumors actually, so they can destroy some particular proteins and get the job done. So, very interesting. Oh, by the way, the people that discovered this, of course, they got the Nobel Prize not too long ago. Alright, so let's assume that we survive those small interfering RNAs or those microRNAs. So let's assume the RNA interference did not play a role and we made it through. So here comes the messenger RNA nicely. So the next step, the messenger RNA needs to go to the ribosome. And as you know very well, here comes the messenger RNA. It's going to attach to the small subunit, messenger RNA. Then comes the start codon, that is AUG. The tRNA brings the first amino acid, that is met. And then when you have small subunit, messenger RNA, tRNA with met, here comes the large subunit and tops it off like a nice sandwich. Well, to do all this, everything has to fit perfectly. Remember, it's all about shape. So now we have a new level where we can stop things. There is a number of proteins that are called regulatory proteins that can actually, see here you have your small subunit, messenger RNA, your tRNA. We are ready to bring the large subunit. Well, not if we have a regulatory protein. These are proteins that actually regulate the process of translation and they can block it any minute. So, if you have that regulatory protein, guess what? That regulatory protein can attach to the messenger RNA here and physically prevents the attachment of the large subunit. No large subunit, no translation. So, another level, how you can stop it. You thought that you were so happy we finally made it to the ribosome, and here comes the regulatory protein and says, no sir. So, Again, nice way that your cells can, using multiple levels, they can block or stop a process because things change in our multicellular bodies. So, well, now, let's pretend now that we made a protein, that messenger RNA made it to the ribosome, everything went well, and we were able to complete translation. What the ribosome makes is really officially called a polypeptide, just a chain of amino acids. It's not done yet. Remember that a protein, to be totally done, it has to fold. It has to, this is what the ribosome produces, just a nice chain of polypeptides, of amino acids forming a polypeptide. So what needs to happen now, we need to start folding, and that's where we have the secondary structure that is determined basically by hydrogen bonds between the R groups of the different amino acids, and you have the alpha helix or the beta pleats. And then when that is organized, when those hydrogen bonds are like that, then you have the tertiary structure that takes into account hydrophilic, hydrophobic interactions, and disulfide bonds, all that because of the R group in each amino acid. So, we have the folding that still needs to happen, and the conditions for the folding have to be right. So if you look at this diagram, a lot of these proteins are going to be made in the ER. Remember that there is that signal peptide, the first few amino acids that allow the protein and the ribosome to move to the ER. Once the protein is in the ER, from the ER it's going to be placed in vesicles, and those vesicles are going to move to the Golgi. And the Golgi is the big organelle, basically, that is going to complete the modification. Any other modifications that need to happen to this protein are going to happen here in the Golgi. What kind of modifications can happen now? First, it needs to fold properly. So that's number one. If it doesn't get to fold properly, all this work was for nothing. So folding. Cleaving. Removing pieces. Sometimes some of the proteins need to be cut up. There are a lot of extra pieces that are signaling molecules that need to be removed. If these pieces are not removed, nothing happens. For example, insulin. Insulin is really produced as a one long molecule. But in reality, before it becomes active, you have to cut it in half. Remove a couple of pieces and attach those two halves together. So all that work to make insulin functional needs to happen. If that doesn't happen, you can make insulin, or in the way it is called really pro-insulin, and nothing happens because it needs to be cut, cleaved, and refolded. So very important. Cleaving. Again, you have to target it for transport. It has to go through this, and the Golgi has to be able to put the right tag indicating where it needs to go. If half the way through your body decides, oh, too late now, we don't need that protein in the membrane anymore, guess what? It's not going to be tagged, and the protein is going to probably be placed in a vesicle and destroyed by proteases. Now, if this protein that is moving through the Golgi here, I'm just giving you examples of things that can happen, is a protein that needs to go into your cell membrane. And a lot of the proteins in the cell membrane are what we call the identity proteins, that tells you what kind of cell you are signaling proteins. All those signals are really carbohydrate molecules attached to this protein. Just like for the blood types, the different groups are because A, B are different carbohydrate molecules attached to the membrane of those red blood cells. So these identity markers that we call, they are basically sugar groups that need to be attached to your protein, and those are attached right here in the Golgi. So that is called glycosylation. I love that name, glycosylation. Remember, anything with glycolysis, glycogen, glucose, all that glue means a carbohydrate, really. So any protein that is going to be destined to go to the membrane needs to be glycosylated in the Golgi. If it's not glycosylated, guess what happens? Absolutely nothing. All that work for nothing. So we can have glycosylation, we can have the cleaving, chopping, basically, pruning that needs to happen. And the one that you also are very familiar with is the process of phosphorylation. You know that many proteins, in order to be working, they need to have a phosphate group attached that makes them basically very reactive. No phosphate, and they are very unreactive, very passive, because the charges are not right. So a lot of proteins are made, but until the moment that they are phosphorylated, that a phosphorus group is attached, they remain inactive, and technically that gene is still off. So again, all these things that need to happen and work beautifully in your body for a gene to become on, to be expressed, to be active. When you think about this, it's just unbelievable that we function and work so well. So many things can go wrong. So there you have it. Now, let's celebrate. Let's pretend everything went well. We have our protein. It's folded or phosphorylated or glycosylated. It's ready to do its job. Actually, some of them get to do their job, and then they need to be destroyed because they didn't need to be around for that long. Or sometimes even before they get to do the job, they get a conflicting signal and said, stop, it's too late, we don't need them. Regardless, we have a whole new field that is called protein degradation, and this would be the last step in the process of gene expression, the last thing that you can do. You make the protein totally functional. Now, let's get rid of it. And to get rid of proteins, they discovered that it's not just, again, magic. Things just don't happen. But your cell has a number of ways to destroy or eliminate things that are not needed. And this leads us to another protein called ubiquitin, because it's ubiquitous, all over the place. Were discovered not too long ago. And this is a protein that is not very big. It's only about 76 amino acids long, relatively small protein that I have here in this diagram represented by these green circles. When your cell doesn't need a protein anymore, either because already worked a little bit and you are done with that process and you need to destroy it to stop the process, or because whatever factor, your cell is going to tag that protein to for distraction. So this is the protein. Oh, look at all those helixes there. Nice beta alpha helixes there. So let's assume this protein fulfilled its mission in your cell and is not needed anymore. Okay? So what your cell does is going to use ubiquitin, this protein, and it's going to attach it to the cell, to the protein to be destroyed. So this is a tag, and this is a tag that says you are marked for destruction. You are going to be totally destroyed. Who destroys it? Another protein. So this is like cannibalism, proteins destroying proteins. Here comes another humongous protein. It's a three-dimensional protein that looks kind of like a barrel called a proteasome. Proteasome. Some means body, the body of a protein. So what happens here? Any protein that has a ubiquitin tag is gonna be surrounded by this proteasome, and the proteasome basically is gonna, it's like a nice grinder, and it's basically gonna totally destroy the protein. And that's the end of it. All the amino acids, basically, all the small peptides are going to be basically destroyed further into amino acids by proteases in the cell. And basically, all the components are going to be recycled. They're going to be picked up by more tRNA to build more proteins. So remember, all these components, when they get degraded, broken up, they are totally recycled and reused. It's the same building blocks that we need to build absolutely everything. So no need to waste anything. So these guys, the proteasomes, actually have the nice nickname of waste disposers. And the ubiquitin is basically your death tag. You're it. Ubiquitin, this is fairly recent. Notice they received the Nobel Prize. Well, we've known about ubiquitin for a long time, since the 80s. But the people that discovered them and studied them and figured out received the Nobel Prize in 2004. And here is a nice representation of what ubiquitin looks like. Nice alpha helix here. Whenever you see these arrows, actually, this is an important point. This represents beta pleats. This is how beta pleats are represented when you have a three-dimensional view of the protein. So this is the tertiary structure of your ubiquitin that includes nice alpha helixes and nice beta pleats there. So, ubiquitin, death tags. So, we just completed basically a walkthrough through all the things that can go right or wrong, depending on your point of view, that can basically turn genes off, basically stop the production of a protein, and is part of what we call gene regulation in our bodies. So just to walk through, here we go. Number one, we can regulate at the level of transcription, and for that, DNA packing around histones is very important. So histone acetylation unravels and turns genes on, and DNA methylation blocks the genes and turns genes off. In addition to that, you have all the job of the enhancers that can activate, basically promote more transcription, or if you have some repressor proteins or silencer proteins, they can block it off and stop it right there. If you make it through transcription, you have all the post-transcription modifications, and you have splicing. Nice splicing. You have the 5' cap, the poly-A tail, and then as soon as you go into the cytoplasm, that I don't have it here necessarily, is the new stuff that has to do with the breakdown or blocking of messenger RNA by small interfering RNA or microRNAs. That happens right here in the cytoplasm. If you survive the attack of the interfering RNA and you make it to the ribosome, then we can have some issues with translation, and we have those regulatory proteins that block translation and nothing happens. Now, if we make it past translation and we have a protein, well, then a couple of things can happen. We might have issues with the processing of the protein, either the glycosylation or phosphorylation or the proper folding, and everything stops right there. Or, if everything goes well, you can make it, you get to use the protein, but sometimes, or most of the time actually, you don't need to have a protein forever around, so we are going to end up with protein degradation that is accomplished by ubiquitin tagging the protein, and then the big proteasome basically destroying the protein. So, I hope with this, none of this is really new to you. You knew most of the components, but this kind of puts the whole picture together of all the many things that can happen that can basically turn your genes on or maintain them on or turn your genes off. And this is what's gonna be happening throughout our life from the moment that we become a zygote until we become this multicellular structure with differentiation in our body cells. So we'll stop here and we'll continue with the development in the next video.